Hi, I'm Mawalagedera. I'm a postdoctoral research fellow from the Australian National Insect Collection here in Canberra. Let me start off by acknowledging the traditional owners of the lands that we stand on today. For us, it's the Ngambri and Ngunnawal people, and I pay my respect to their elders past, present, and emerging. So the topic that I'm talking on today is screening Hymenoptera insect collections to putatively identify bioactive metabolites. Hymenoptera species are diverse in natural products, and many of them have antimicrobial properties. As an example, there are apitocin-type peptides derived from bees, wasps, and hornets that has recently been studied, and they have been screened against mammalian cells, and they have no adverse effects. In addition to that, there are well-studied piperidine alkaloids that derived from red ants, and many of the Hymenoptera also have surface hydrocarbons which have antimicrobial properties in their colonies. So when you're using these natural products from Hymenoptera collections, screening them efficiently is crucial to identify compounds that are useful to humans or other animals. Therefore, we think that the preserved natural history collections provides a good source of screening these natural products. Therefore, the objective of the research that we are doing was to identify the potential of using these preserved insect collections for the purpose of biodiscovery. So in order to do this, we have an initial biodiscovery pipeline that we have developed. This starts off by selecting several taxa from the collections, and we basically homogenize the tissue and we do a biphasic extraction, where you're essentially homogenizing the tissue with methanol and chloroform, and then you're separating the extracts into 3 layers, which is the upper polar phase, the middle protein phase, and the lower non-polar phase. Then you have the potential of using 3 of these phases to do a chromatography mass spectrometry, which essentially separates the metabolites into ions. This can be done either using gaseous phase in gas chromatography mass spectrometry or liquid chromatography. In this case, we have done gas chromatography mass spectrometry using the polar upper phase of the insect extracts. Then the resulting chromatography information that we derive from GCMS could be imported into mass spectral databases, which are either vendor-based ones or open source databases, or you can use a large repository of mass spectral information, such as global natural product social molecular networking, to identify those chemicals coming out from the extracts. This is quite similar to having a DNA sequence and you put it on NCBI and you're getting reference sequences. And this screening could be done in 2 ways. One, the targeted analysis, where you're trying to identify a set of known metabolites to which you already have the chromatographic reference information with you. Or in the other analysis, you can do it in an untargeted way, where you're essentially trying to identify all the metabolites in those extracts. And in an untargeted analysis, therefore, you would have a set of unknown metabolomic data as well. Explaining this further, so the chromatogram here would be the output from a GCMS, and then you process it further, and then you get peak data with mass to charge ratio and abundance of these compounds. And then you have your reference vector, which are the ones in online databases or vendor-based databases, and you match these 2. And in GNPS, what they do is they match this number of peaks, and then they develop a sort of a molecular network where they have numerous clusters, and they give a similarity score between these 2 spectra. Therefore, in a molecular network, you would have numerous clusters, and a single cluster would correspond to a particular metabolite or a chemical class. So moving on to the sampling methodology, from the Australian National Insect Collections, we picked 2 families, Pompilidae, Typhidae, and then we sampled specimens which were in the pinned collection with different ages, and then some from the ethanol stored collections we sampled both the ethanol stored tissue as well as the corresponding storage solvent. It was the same for the Typhidae. For the Typhidae, we had the Diama bicolor, the blue ants, and for the pompilids, we used Eplaniceps. And then we had the pin tissue, ethanol stored tissue, and the storage ethanol. And all of these tissue samples, as well as the corresponding solvents, went through the prior mentioned biodiscovery pipeline. We took the polar phase and we analyzed them using gas chromatography to get the metabolomic identifications for the data. The resulting data or the peak information then can be exported onto different software to do analysis of the peaks. And then we sort of exported these data into a web repository called MetaboAnalyst. And you can align the data and do different statistical analysis. So in order to see how the peak data is shared between treatments, we did a partial least square discriminant analysis. And here you can see that both the pompilids and the different treatments clustered within their 95% confident interval. You could see the ethanol stored tissue, the ethanol solvent, then the pin specimens of the pompilids. Here the ethanol only again, ethanol specimens, and the pin specimens of 2 different ages. Next, we also wanted to see how much of the metabolomic or the chemical information is shared between different treatments. For the Hymenoptera, the pompilids we saw in the targeted analysis, there was more than 95% of the chemical information shared between treatments, regardless of their preservation conditions, age. For example, for the PINT 2018 and the 1965 specimens, we saw that there was 168 common chemical profiles between the two, whereas only about 2 and 1 were unique to the treatments. And it was quite similar for the ethanol stored specimen and its storage solvent. However, when we went to the untargeted analysis, reminding you that here we have both identified chemical data as well as the unidentified chemical information, there the shared information was down to about 50%, where a large portion of chemical information was unique to different preservation age specimens, as well as the ethanol specimen and its corresponding storage solvent. And the situation was quite similar for the Typhids as well. In the targeted analysis, we saw that between the ethanol stored tissue and its corresponding solvent, it was about 86% similarity, but in the untargeted analysis, it was down to about 50%. Then we wanted to explore some identifications for these compounds. And I'm presenting here the results from the molecular networking that I was talking about. Here you have the huge molecular network and you have different clusters. What we did here was we went back to the literature and tried to find previously characterized chemicals from the blue ants and also the Euplanus notable species that we used. However, there was not much information for the chemicals in the Euplanus species, but there was some information for the blue ants, and we were able to pick out 2 clusters that gave us compatible putative identifications. One was the serotonin-related cluster for the blue ants, which results in intense pain during the bite. And then there were the long-chain cuticular hydrocarbon-related clusters, potentially from the Hymenoptera exoskeleton or their surfaces. Then we further explored the serotonin-related cluster from the Typhidae. This is the network identification that we got for serotonin. And then we did the similar Venn diagrams to see how much of this information was shared between different treatments for the PIN 2018, 1965, ethanol stored tissue and its solvent. And we saw that about 35% was shared, whereas some of the peaks related to the serotonin cluster were unique to different treatments. So moving on, what do we gather from these results? Due to the difference in the amount of shared metabolomic information between the untargeted and targeted analysis, we can conclude sort of that there is a chemical degradation during the preservation process, which could be due to the process of some metabolites leaching into the storage solvent because they've been stored in ethanol for a very long time, and also due to the reason that the actual chemicals are degrading, and again, or due to the degradation, the detectable concentrations of those chemicals are lower in the long stored tissues, and they might be below the thresholds that we set in detecting these chemicals in our analysis. Therefore, if we are using the natural history collections as an initial screening point for bioactive metabolites from Hymenoptera, the ideal condition would be to screen lots of specimens stored under different conditions and ranging from different ages if we are to get a holistic view of the chemical profiles in the natural history collections. So moving on, we want to expand this research into our natural history collection more and collect as well as fresh flash frozen specimens and their corresponding long-term stored preserved specimens from different ages, different storage conditions, and screen them all together and have a holistic view of how much of that metabolomic information has been degraded over time. And then we want to ID bioactive metabolites more robustly, and for that, we want to use past references and then have their reference material from manufacturers and then match the chemical information and then have robust identifications. Also, we hope to screen the collection much more systematically so that it would allow us to sort of do phylogenetic prediction of bioactivity using methods such as mantle test or D-matrix or phylogenetic eigenvector mapping. And we are also thinking of coupling antimicrobial testing, actual antimicrobial testing of those extracts to be more certain of their biological activity. So moving on to the end, I would like to extend my thank you to the Hymenoptera Lab at Australian National Insect Collection, especially to our project lead, Juanita Rodriguez, Vidisha Patel, Daniel Dashwesky, Olivia Evangelista, and Leonardo and Madeline. Also to the Joint Mass Spectrometry Facility at Australian National University, Dr. Adam Carroll, who helped us in doing the GCMS. Thank you very much for your time, and we are open to your questions.
Thanks, Sundara. What a fascinating talk. It was metabolomics rather than genomics, but I think it fits right into this session. Do we have any questions first up? I'll ask a question which is a little bit, I think it's a little bit naive, but I'm still really interested. So if you have an idea of the compounds that are in the historical specimens, how can you use that information with regard to present day species? Are you trying to use that information to infer what taxa might be of interest for those particular types of compounds?
Yes, that's one of the next steps of our research as well. We want to get the fresh specimen and then also the preserved collections and see the chemical degradation as well. Because in addition to storing over time, a lot of abiotic stresses also affect the expression of chemicals and chemical changes due to the evolutionary arms race and herbivore stress, citrus stresses, and the co-evolution between plants and insects is also there. Therefore, we do want to screen both the current specimens as well as specimens from different collection events to get a broader idea of what has been changing.
Okay, cool. Thanks so much. Okay, we're going